So good morning. Hopefully at this point everybody has figured out how to get to the ebook, right? And is maybe getting close to figuring out. Okay. All right. So see me after class. Do you have time to do that? Okay, perfect. Anyone else having difficulty with that? Because I went over it last time. Remember I recorded it. Vanessa was kind enough to get up and get onto her account and everything like that. Pretty soon we're going to start having assignments, right? They're not up yet, but that's coming soon this week. Our exam dates are set. You'll notice I added a new page this morning. This is animation, video, and videos for Module 1. One of them I want you to watch for homework. The good news for this one, Vanessa, it should have correct captioning, I'm hoping. We'll see. And then under objectives and PowerPoints, and then of course, this is where I post the lectures after class. Remember, I'm posting them to YouTube, so if you subscribe to my channel, you probably get announcements before it even shows up here in Canvas. And I usually in Canvas date them here for you, so you know what day, if you missed class, what day we covered what. So those are there for you. Remember, everything is pretty much in modules. And so the other updates that I made today, this morning, and this is kind of small, huh? Let's make it a little bit bigger. So we should hopefully finish up this first set of objectives. I haven't made any changes to this. I think it had some out-of-date material. I'm probably not going to put chapter stuff there because it'll probably change with the new edition of the book, as it always does. So I started putting notes here underneath the PowerPoint. So there's some information in this PowerPoint that we've been going over that's not covered in your book, right? But you're responsible for it. For Koch's postulates, how many of you guys actually read the last section of Chapter 1? Okay, and when you read about Koch's postulates, it told you to refer to another chapter, did it not? Let's see, who's the super reader? Who went to the next chapter? If you looked at my PowerPoint, you should have seen that I go into more detail than they did in Chapter 1, right? Which would require you to go to Chapter 18, Section 4, like the book says. So I'm going to try and give you guys these hints ahead of time, right, from now on. So that's what I've already done for the next PowerPoint, is that, again, it's going to contain some material not covered in your book, but it's not tough stuff. You're going to need to read Appendix B. It's 4 pages, y'all. You can handle it. And you need to read Section 2.4. You don't even need to read all of it. You need to start where it says Variations in Bacterial Envelope. And then you also need to read the Toolbox 2.1. In addition to that, I want you guys to watch the Gram stain animation. And I even put the link here for you guys to that page I showed you a moment ago, so you don't have to go back to modules. So this is the Gram stain animation. And this is a YouTube. So you can click out even to YouTube. Let's see. And like I said, I'm hoping the captioning is correct. I'll watch through it and let you know. But at least watch it, right? Because there's a lot of visual. Anyways. Okay, does that make sense to you guys? So that's kind of your homework for next time, right? Is to read. And then the PowerPoint's already there if you want to download it. Here, I'll just hit my back button. So there's our second PowerPoint right there. And this is the objectives list, which are, of course, covered in the PowerPoint. It's just a quick list. You'll notice it's only 3 objectives, but it's a lot of material. Any questions on that? So I'll continue to do that as we progress through the semester. So let's go back to our, I should have opened it, first lecture. So we've talked about in general importance, right? And they go into a lot of that in Chapter 1. Hopefully at this point you guys have read most of Chapter 1. He's a really good writer. I actually enjoy reading this book. I hope that's true for you guys too. So these associated pathogens, of course, right? This is content that's in the PowerPoint only. Remember we said this was going to be kind of like matching. So we ended with that, and now we're going to start the next objective. Which really, what do you think of when we think of microbiology? How would we define this field of study? And really when we do that, it's very large and encompassing. And a lot of the techniques are used by so many disciplines. So we're going to also look at some of those general techniques today. And they kind of fall under our understanding and definition of microbiology. So when you think micro, you think what? What does micro mean? Small. What does biology mean? Study of life, right? Ology, any ology is the study of. Bi is the study of life. Well, that gets kind of tricky when you start talking about microbiology, because we do study some things that most scientists don't consider to be alive. Does anybody know something that's actually submicroscopic? You know, the standard light microscopes you use in your microbiology lab are not going to see these infectious particles, but they can cause serious illness for us and even death. What do we consider that's not living? You can't see it with a light microscope. It's too small. Viruses. And you guys are going to learn about some other entities, right, that we'll talk about that you probably never heard of before, like viroids and viceroids and satellites and purons or pyrons, commonly also referred to as just infectious proteins. So a lot of times we don't even say like the study of microorganisms anymore. We say the study of microbes. Microbes is a more general term that includes things like viruses and all those others I just mentioned. And we'll, cover a whole topic of those in Module 2. So we're just going to introduce them here, and then we'll go into more detail later. So normally these are things we can't see with the naked eye. Normally we would say microorganisms. Nowadays we say microbes. And then of course we talk about the techniques that we use to study them. And for the most part, right, always be careful of true and false statements that say all or only, right? Is there anything really in science that everything fits into that one category? Not really, right? There's always exceptions, right? So believe it or not, there's some microscopic organisms that if you've got really good eyes, you could probably see them. Not in detail, but you could see them. For the most part, they're relatively simple in their construction, but as our techniques improve, as you guys read for homework, like the high-powered microscopes we have at our access nowadays, I mean, we're learning so much more. When I was sitting in your seats as a microbiology student and undergraduate, some of this stuff that I'll talk to you guys about hadn't been discovered yet. Granted, I'm getting older. That was like 20 years ago. That still doesn't, that's not a long time ago, right? Not relatively. So we've learned a lot in a short period of time due to the techniques that we have available to us today. So for the most part, they obviously don't have differentiated cells and distinct tissues, like multicellular organisms like ourselves. But there are some that are microscopic. Anyone ever look at rotifers? Search rotifers on Google, right? They're pretty amazing microscopic little animals. They're so unique they have their own phylum. You can see through them. You can see their digestive tract. Right? If you go take a sample of pond water, you might run into one. They're pretty awesome little guys. So again, these guys have distinct tissues. They have a digestive tract, right? But yet they're microscopic. So again, there are some exceptions, but for the most part, most of the things that we look at that are microscopic are pretty simple in terms compared to obviously things that are much more complex, like our cells. Make sense to you guys? So this is a really cool website that one of my students in the past turned me on to. It's actually a genetics website at Utah. And of course, it says it can't find it. I don't think I added it to my page of animations and videos, but one can only hope, right? Nope, of course not. All right, so we will, what is it? Cell scale Utah. Maybe that'll be enough information to, yes, find a more current link. So this is interactive. The one thing I like about this too is down here, it's kind of grayed out. I don't like that. But it gives a different metric system measurement. So most of you guys have measured in meters and centimeters and millimeters, right? Any of you guys ever heard of micrometers? No, not until you get to looking at stuff under the microscope are we looking at something that, in comparison to a millimeter, which you can see on a centimeter ruler, those little lines are millimeters. These are 1,000 times smaller than a millimeter, right? So in relationship to a meter, they're -6, so 1,000,000 times smaller than a meter. And then nanometers, angstroms, and pinkometers, right? So we're getting really, really small. I really don't like that this is highlighted dark. Makes it really hard to see. I don't think that improves it at all either. Gonna have to send them an email. It didn't used to be like that. But so when we look at this, what they've done is, like I said, this is interactive. So this is in relativeness, right? The size of a coffee bean is about 12 by 8 millimeters, right? This is something we can easily see. You may even be able to still see, too, of course, a sesame seed or a grain of rice. Right, and this is Times New Roman 12 point. So as we move the bar, we can zoom in. So in comparison, a grain of salt, right, you would really need a microscope to see the detail, right, of the cube structure of a grain of salt. Right, it's 0.5 millimeters, although apparently some people can see stuff that small. I'm not one of them. Right. So we really need the aid of the microscope when we start getting into micrometers, right? So something that's 500 micrometers, an amoeba. So if you're in lab this week, that's one of the things that you'll look at is a single-celled eukaryotic protozoa, right? Again, that you could find in pond water. Same thing with paramecium. You've probably looked at these guys before in a biology class, right? The human egg, which is the largest cell in a woman's body, because it contains all the organelles, right, and everything for that developing baby. All sperm is mobile DNA. It has a few mitochondria, but mostly you get your mitochondria from your mom. Your skin cells, right, still relatively large under a light microscope as compared to much smaller things like baker's yeast, something we'll also look at in microbiology lab. These guys are going to look like tiny little round oval specks under the microscope. You're going to be like, that's it? Right? Poor old E. coli, Archaea coli, is actually smaller than a mitochondria in a eukaryotic cell. Right? The lysosome, an organelle found in a lot of eukaryotic cells, is quite large even compared to other things like viruses, which you're not going to see in the lab under a light microscope. These are just too small. Mm-hmm. Yes, you can absolutely survive an E. coli infection. It depends on what strain of E. coli, though, you're exposed to, right? Bacteria do differ, not necessarily at the species level, but differ quite a bit at the strain level. And some of them have acquired stuff that we're going to talk about towards the end of this lecture, abilities to be pathogenic, so cause disease, make you sick, possibly kill you. So you've probably heard of the strain E. coli 0157. That is a very pathogenic strain of E. coli that we're going to talk about throughout the semester. But usually the standard stuff in your gut, if you're not in the bathroom of the hospital right now, is not the bad guy. So viruses are teeny tiny, right? And we'll talk about different ones. You'd need something like an electron microscope that you're going to read about for homework. We could start getting down to the molecular level. Yeah, believe it or not, we have microscopes that can go this tiny. You could see complex molecule structures like ribosomes, which all cells have because what do they make? Proteins. And they're made out of protein and what else? What are ribosomes made out of? It's a special type of RNA called rRNA. Guess what the R stands for? Ribosomal. Right? And it reads another type of RNA known as messenger RNA. This was in chapter 1, by the way. It's a review of biology. Thank goodness someone said yes. Yeah, because we're going to be talking about this stuff. Okay, so there's the transfer RNAs too, another RNA that would actually transfer the amino acid to the ribosome messenger RNA complex. And these are my favorite proteins, antibodies, right, produced by our immune system that give us immunity to different types of pathogens like E. coli. Hemoglobin, obviously very important. And then an individual amino acid like adenine or single sugar like glucose or methionine, another amino acid. Good old water, which everyone knows is made up of what two elements? H2O, right? Don't get H2O2 though, right? What's that? Hydrogen peroxide. There's a chemistry joke where a guy walks into a bar, says, I'll have H2O. The other guy says, I'll have H2O2. They give him hydrogen peroxide and he dies. Right? Even chemists have a sense of humor. At the core of it all is what element? Carbon, right? We have carbon-based life forms. All organic molecules have a carbon backbone, right? It all starts out with the carbon atom. This is why we can do what? To figure out how old something potentially was. When was it once alive? Carbon dating, right? Because we know the decay rate of this molecule over time. And so we can estimate, based on that decay, how old that substance was when it was alive, right? How long ago was it alive? Make sense? So I thank my student for turning me on to this awesome site. And apparently we need to fix our link, though, in our PowerPoint. So instead of doing it there, I'm going to add it here. I'm going to put it at the bottom. You can tell I didn't check it before class, shame on me. So if you want to go look at it again, show your friends, your family, your kids, that link should work. Let's check. Yay! So the next we talk about, and again, these are definitions that have been around for a long time. And as we're discovering more and more, scientists are starting to question, are there really these two groups? Because we're seeing, crossover between the two groups. So how can we separate them into two groups if there really isn't two groups, right? But as of right now, there isn't enough evidence to abandon these two cell types that we commonly refer to organisms being. So we have what's referred to as prokaryotes and eukaryotes. So by definition, anyone know what pro means? What does pro mean? We took medical term. Come on, y'all. You know this stuff. P-R-O, pro. Before, right? Karyote actually means kernel. So when scientists were looking at cells under the microscope, they saw this round thing inside of some of the cells they were looking at. What was that kernel? What was that round thing in the center of that cell that they were seeing? What do we call that nowadays? The nucleus. And we know that this is found in eukaryotes. Eu meaning true, karyote referring to that kernel, that nucleus that we see today. So by definition, if prokaryotes are before the kernel, do they have the kernel? No, so therefore they lack what? A nucleus. And that's the major defining characteristic between prokaryotes and eukaryotes.
I have a question. On eukaryotes and prokaryotes, can that be spelled both ways? Because it was with a K and now it's with a C.
Yeah, and I purposely left it messed up like that in my lecture so you guys would ask me that very question. Some people spell it with, some countries spell it with a C, some spell it with a K. Both are correct and acceptable. So just like my name, Erica, some people spell it with a K, I spell it with a C. I was supposed to be a boy. I had to add an A. Oops. So, hopefully you read about Carl Woese. So again, when I was sitting in your guys' seats, scientists hadn't agreed yet, hadn't made it into your textbooks. I didn't learn about the 3 domains. Right? It wasn't until 1990s, right, that it really became accepted and then transitioned into your textbooks by the scientific community. So among those, right, so if we need to describe the very basic differences between prokaryotes and eukaryotes, and the very simplest is that, does it have a nucleus? Does it not? Their morphology and their distribution among the various kingdoms or domains. And so the 3 domains that Carl Woese came up with are Archaea, bacteria, and Eucarya. As the name Eucarya implies, guess what cell type all those cells are that fall into the domain Eucarya? Eukaryotic. The other 2 by default are prokaryotes. Bacteria and Archaea are prokaryotes. So as we've already defined, prokaryotes lack a true membrane-delineated nucleus. So another question for you, back from your biology days. What very important molecule of inheritance is found inside the nucleus? DNA. What does DNA stand for? It's an acronym. Deoxyribose nucleic acid. So deoxy means the sugar ribose is missing what? An oxygen that a normal ribose would have, sugar. You have a nitrogenous base and a phosphate group. For each one of the molecular components, those nucleotides that make up DNA. So for eukaryotes, that's found in the nucleus. Prokaryotes, no nucleus. Do they have DNA? Yeah. All cells have to have DNA. It's the genetic code. It's just not encased in membranes. It's in the cytoplasm. We don't compartmentalize it for prokaryotes. We don't stick membranes around it. For eukaryotes, on the other hand, again, that's a defining characteristic of a eukaryote. It has that membrane-delineated nucleus with the DNA inside. And it usually is more complex morphologically and larger than prokaryotes. Notice we said usually. There are obviously some exceptions. Always, yeah. It's science. It's life. So back in the day, and they talk about this in your book, give you a little history of classification. At one time, we used the 5 kingdom scheme. You had kingdom Monera, Protista, Fungi, Animalia, and Plantae. With microbes in the first 3 kingdoms, right? Plants and animals are, of course, macroscopic. They're not microscopic. Monera, what type of organisms would you think would be in that category that we talk about today? So they're microscopic. What domain even? So we have bacteria, Archaea, and Eucarya. Which modern day domains do you think were lumped into Monera? Good morning. I just gave you 3 to choose from. Bacteria, Archaea, and Eucarya. Bacteria and Archaea were lumped into Monera. Protista would be like that amoeba we talked about, the paramecium. Fungi includes yeast and mold, so that baker's yeast, as well as multicellular, macroscopic things like mushrooms, right, fall under the fungi category. Then, of course, Animalia, there's no confusion there, or Plantae, right? The 3-domain alternative was based on ribosomal RNA, because as we said, all living cells must have ribosomal RNA, must have ribosomes. You can't make proteins without them, right? The flow of information within a cell goes from DNA to RNA to protein. Was it always like that? What do scientists think it actually started out with? Which molecule was the storage of information first, they think? So DNA, RNA, or protein? RNA. They believe it started out as RNA. But again, all living organisms, all cells, even if you're an organism that's one cell, you have ribosomal RNA. So Carl Woese started sequencing ribosomal RNA sequences from a multitude of different organisms, microscopic as well as macroscopic. And he found these 3 divisions kept happening, right? That he could group them based on similarities, and he ended up with 3 groups. He named these groups bacteria, Archaea, and Eucarya. Sometimes it's also called eubacteria, for again, true bacteria. So this is a representation, a small representation of the tree of life. Somewhere at the root there may have been a common ancestor, and then there was this split that happened. Even still, it might not even be a clear split. Because even if you were to say, look at our family trees, do we have some crossover? Yeah, they call that inbreeding, right? Where cousins marry cousins and the trees merge, right? It happens, right? And the same may have even been true for organisms' life on Earth, because it's been billions of years, right? Of course, where's our genus? Anyone know what our genus name is for humans? Homo, right? So we're highlighted in red. And of course, again, we're eukaryotes. And then you probably don't recognize anything in these other branches, right? But hopefully by the end of this course you might recognize a few. I can't even, oh, mitochondrion, yep. Escherichia is on there, right there. Bacillus, might be one you've heard of. When we start get to the Archaea, notice some of them don't even have, names, they just have groupings based on different characteristics. There's good old Giardia. Remember that one is what? What type of organism? It's protozoa, right? And it's eukaryotic. Unlike most of the diseases we talked about last time were bacterial or not even living, they were caused by what? Infectious agents we know as viruses. So Carwos is one of the important guys to know, right? He came up with the 3-domain system that we use today. So you may have learned when you were a kid the classification system of kingdom, phylum, order, I can't remember. So guess what? I learned the saying when I was younger. It was called, Kevin, please close our front gate slowly. How many of you guys know that saying? Other people know some king one. You guys know that one? King something or other. Okay. All right. So Kevin, K for kingdom, please, phylum, close, class, our, order, front, family, gate, genus, slowly, species. But I got to change my sentence, right? Because what do we got to put before kingdoms now? What comes before kingdoms? What do we call bacteria, Archaea, Eucarya? What's this classification? Domains. So, dear Kevin. I say, damn, Kevin, close our front gate slowly. So I have a bad mouth. But dear Kevin would work too. As you guys get to know me, you'll notice that I'm a big fan of tables for organizing information. They're really good, right, at looking at similarities and differences between different categories. So for bacteria, cell type, remember what are our two choices for cell type? Prokaryotes and eukaryotes. So what are bacteria? Prokaryotes. Archaea? Prokaryotes. Eucarya? Eukaryotes. That one's easy, right? Probably won't put that one on the test. All right. Cell organization. So there are some organisms that a single cell is an organism. And for prokaryotes, they're single cells. So bacteria are unicellular. Archaea are also prokaryotic, so therefore they are also unicellular. Eukaryotes. Can they be unicellular? One cell. Uni one. Yeah. Give me an example. Amoeba, paramecium, Giardia. All single-celled organisms. All eukaryotic. Most of them have a nucleus you can see under the microscope. Are eukarya also multicellular? Yeah, plants, animals, some fungi. So this next one I've kind of modified a little bit from in your book. I really want to focus in on one of these defining characteristics of one of these groups. One of these groups, when they have a cell wall, it's made typically out of a substance known as peptidoglycan that you're going to learn about as we start reading and going over the gram stain. So anybody know which one of these we gram stain? Bacteria, Archaea, or eukaryotes? Bacteria. So they're the only ones that contain peptidoglycan. You can stain any cell using the gram stain. But particularly for bacteria, this is going to give us some information as it relates to their peptidoglycan layer. And because of that, we're able to divide bacteria into two major groups. Ones that stain what we call gram-positive, they stay purple. And ones that do not stay purple and get stained with the other dye and come out pink, they're known as gram-negative. This is a result in a gram staining procedure. They're either positive, stay purple, or negative, pink. But doing this procedure tells us something about that peptidoglycan layer. I'm not going to tell you yet. I'll let you read about it and watch the video. So these other domains, do they have cell walls, some of their organisms? Yeah, Archaea have cell walls. Some eukaryotes, like plants, definitely have cell walls, right? Fungi have cell walls. Are they made out of peptidoglycan? No, because that's unique to who? Bacteria. Okay, membrane-bound organelles. What are those? Anyone can name an organelle. Mitochondria. The nucleus. Chloroplast. Golgi apparatus. Remember these things? Who has these? What cell type? Just eukaryotes. Where can you find bacteria on this planet? Everywhere. All environments. What about Archaea? Probably never even heard of them before, right? These guys can live just about everywhere, but they actually prefer extreme environments. It was when we started really looking at really cold and really hot environments, really salty, really acidic, really basic, that we start discovering these organisms. Eukarya, just like ourselves, are you going to find us everywhere? No, right? If it's too cold or too hot, we're not going to make it. So for us, it's pretty much everywhere that's not considered extreme. I didn't understand or hear. Oh, okay. All right. So in the domain Eukarya, they're all eukaryotic cells, as we said. They contain plants, animals, and eukaryotic microorganisms. So we start looking at protists, which include your unicellular algae, so things like euglena, spirogyra, protozoa like amoeba and paramecium and giardia, slime molds and water molds, which honestly I can't give you a single example of. Never really studied them. Pretty cool creatures though. And then fungi, most of us are aware of molds and yeast, right? And then of course mushrooms, which are actually not microscopic. Most are larger than eukaryotic cells. Again, some of them can be pretty tiny, and vice versa. For viruses, these are acellular. This is really important. A means, in this case, that prefix, are viruses cells? No. They are not a cell. A virus is not a cell. Acellular means without or not. So they're not cellular. They're not a cell. They don't have all the components of a cell. They have some components, but not all. They're the smallest of the microbes. Notice the term microbe as opposed to microorganism. They're typically 10,000 times smaller than a typical bacteria. And as we know, they can cause disease and even cancer. So Antony van Leeuwenhoek, do you guys remember what he saw in his poop? What organism that's on our selected diseases list? It's a protozoa. Dead giveaway, there's only one on our list that's a protozoa. Giardia. So I have dates on here, right? That usually freaks students out. I don't want you memorizing dates. I have these dates here to give us a frame of reference, to know how long ago this was. It's a really long time ago. Like, I can't even fathom this, right? 1632 to 1723. He is usually agreed upon as giving credit as being the first person to observe and describe microorganisms accurately. So things like bacteria and the Giardia that he saw in his own poop. How do we know this? How do we know he did this? He documented it, right? He wrote letters to the Royal Society in London. He did drawings. And in one of the textbooks I was reviewing recently, apparently he didn't even do the drawings himself. He hired an artist. It's like, okay, they're not that complicated.
I have a question on viruses.
Mm-hmm.
You said it doesn't have a cell.
It's not a cell.
It's not a cell. Okay, so what is it, a living organism?
Most people do not consider a living organism by that criteria as well as other, the fact that it is not a cell. It's not the basic unit of what we consider life to be. It also can't reproduce on its own, can't metabolize on its own, it can't respond to the environment on its own. So here's Handsome Devil. That's back when they wore wigs. Here's his drawings. Here's what they believe some of his early microscopes may have looked like. So in this drawing, notice there's figure B. Here is the figure C. Then a dotted line that goes boop, 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 boop over here to D. What are they trying to denote by this drawing? It moved. It moved. And then most people, when they look at figure E, they're like, oh, it's a semicolon. No, he saw two round organisms. And unfortunately, it looks like a semicolon. Right? Two dots. Others were what we commonly refer to as rods, or a little bit of a curve to a rod, or a wavy rod. We'll talk about the specific terminology we use to describe these basic shapes that bacteria and Archaea take on in Module 3. Lecture 3. So, spontaneous generation. I know I just have a few minutes, so I'm just going to introduce this one, and unfortunately we'll have to come back to it. But still read on microscope stuff, because we will finish this and get on to microscopes, I pray, on Wednesday. Don't pack up. I'm not done. I want to introduce this topic. I have 3 minutes. I'm going to use them. Get used to it. Okay. I'll try not to go over. So, spontaneous generation. What does that mean? Comes from nowhere. Poof, out of the air, right? Before scientists really did some of the experiments that you'll read about, right, especially Louis' experiment, Pasteur, they believed that stuff could just come out of thin air. Right? They thought if you let meat lay out, it turned into maggots. We now know that's not true. What happens? Where do the maggots come from? What lands on that meat? Flies. What do they lay on that meat? Eggs. What do those turn into? Maggots, the larval stage of a fly. Why did she do that? Because what's the meat? It's food for her babies. She put her babies right there. Okay, you're all set once you hatch. I get it set for you. Yeah. Smart, Mom, huh? They don't have to go anywhere. It's right there. But those eggs are teeny tiny, right? If you're not very observant individual, you might not have seen those. And then for microorganisms, right? Nowadays, you stick apple juice and it gets pushed to the back of the fridge, right? Apple juice is usually translucent. You can see through it, right? What happens if you forget about it, right? And a month later, you dig it out from the back of the fridge and it's all cloudy. Who's drinking it? Nobody's going to drink it? Why not? It's apple juice. It's cloudy apple juice. Is it supposed to be cloudy?
No.
So what's that cloudy stuff? Microorganisms that have grown to large numbers, right? And could those potentially harm you? Absolutely. So we're going to skip that, right? We know this, right? We know today, back in these times that we're going to talk about, it just poof, out of the air. Bam, spontaneously generated. Right? Okay, see you guys next time.